Today, I'll talk about Mendel's laws of inheritance. Before going to Mendel's laws of inheritance, we have to understand what is heredity and what is variation. Heredity is what you receive from your parents. It is how similar you look like is heredity, while how dissimilar you look like is variations. Variation is individual you, whereas heredity is your parents. What you get, the characters and the phenotypic and the genotypic characters that you get from your parents is heredity, and variations is individual to you. They are the differences, that how different you are from your parents. So here it is interesting to note that heredity was given by Mendel, whereas variations were explained, or rather, no, they were given by Charles Darwin. Both of them, they were contemporaries. In fact, they studied during a similar period of time, but they did not know each other. Had they known each other, maybe they would have taken their understanding much further in those days, but they did not get in contact and they did not know about each other's work. Now, coming back to Mendel. Mendel, his name is John Gregor Mendel. So he studied in a monastery and then he applied for a university for teaching position. The exam which he could not clear, so he came back to his monastery and he started some research work. He started hybridization experiments on a plot of land that he had near to his house, and he started hybridization experiments on Pisum sativum, the pea plant. The Pisum sativum now is known as central law of genetics. So he did some experiments from 1956 to 1965 for about 9 years, and he published the reports in annual reports of Natural History Society of Brann. Now, after he gave his reports and after he published his reports, so there it caused very less interest among the scientific peers of that time, and it was slowly his reports were lost into obscurity until they were rediscovered by three great biologists: Carl Correns, Hugo de Vries, and von Teschemark. So they called them. They took out his reports and they organized them in proper fashion, and they called them Mendelian laws of inheritance. So Mendel started study on Pisum sativum, as we just talked about. This Pisum sativum is a bisexual plant and it undergoes self hybridization through itself, and that is one of the biggest plus points that Johann Mendel had while he selected Pisum sativum. Also, Pisum sativum has qualitative characters and it does not have quantitative characters. What are qualitative and what are quantitative? We'll come back to that later. So all these things, it was by pure luck that he chose Pisum sativum. Pisum sativum was very uncomplex, very genetically it is not very complex, and Gregor Johann Mendel himself had some background in science and mathematics, and that is why he was able to bring down to genetic ratios. He combined both his knowledge of science and mathematics to arrive at the whatever the loss of Mendel that he gave. So he studied 7 characters in pea plants. The first one is stature. Now, each character, these 7 characters, are represented by 2 factors. So now we call them genes, while earlier they were called as factors because they were called factors by him. So 2 factors represent each character. So 7 characters are represented by 14 factors. So the characters are: first, stature. So it is given by tall and dwarf. Seed color, yellow and green. Seed coat, round and wrinkled. Position of the flower is axial or terminal. Color of the flower is purple or white. Pod color is, the pod is a fruit. The color, that is green or yellow again. And pod shape, that is inflated or constricted. So these are the 7 characters and the 14 factors. He studied them until so first he started hybridization. So he put all the plants and he selected only tall plants and put them in one garden, one piece of land. Then he took all dwarf plants and put them in another piece of land. and he started breeding among themselves. And till 8 or 9 generations after that, he could not get any more dwarfs in all tall pea plants in that plot, and he could not get any more tall plants in all dwarf pea plants. So that way he obtained pure breeds. After he obtained pure breeds, he then crossed among themselves between two similar factors, like the characters. So tall was crossed with dwarf. The yellow seed color was crossed with green seed color. The wrinkled seed coat was crossed with round seed coat, and like that. And he noted his experiments. So since he studied only one type of character, these are called monohybrid studies. Later, he also studied two characters at a point of time, and they called them dihybrid studies. And then three also he studied together and called them trihybrid studies. So initially he studied monohybrid studies, and these monohybrid studies, after studying, he gave three postulates. The first one was law of unit factor. Second one was law of dominance and recessive. The third one was law of segregation of gametes, or also called as law of purity of gametes. After he studied dihybrid and trihybrid experiments, he formulated the fourth law, that is law of independent assortment. So we'll come back to all the laws, then we will understand what these laws are and what their criticism is. So the first one was law of unit factor. So law of unit factor is law of unit factor. This is homogeneous, and this has to do with homogeneous and heterogeneous. As I said, every character is represented by two factors. If both factors are similar for that character, for example, if you have two talls or two dwarfs, it is called homozygous. Now, since that is alleles, so he called them as factors. So two factors, both of them, if they are tall, then it is homozygous tall. If both of them are short, then it is called as homozygous dwarf. And if a plant contains one tall and one dwarf character, or one tall and one dwarf factors, the plant will still be tall, but it will have only half of it is tall and the other half of it is dwarf. So these are called heterozygous. The heterozygous plants give out two types of characters, that is two types of factor, that is one tall, the second is dwarf. So each of them, they go into gametes, and the tall factor, when it goes into one gamete, it becomes tall gamete, and the short dwarf character, if it goes to another gamete, then it becomes dwarf gamete. So that is what is law of unit character or unit factor. So the second law, we'll come back to second law. The second law is law of dominance and recessive. So what is law of dominance and recessive? If the first generation, so that is F1 generation, so he had two pure characters. For example, he had all tall plants and he had all dwarf plants. When he breeded them among themselves between one tall and one dwarf, he got the first generation was only tall, and this tall character he named it as dominant character, because this first filial generation, that is F1 generation, had one character from the tall plant and the other character came from the dwarf plant. But in spite of having one tall and one dwarf, the plant itself was tall. So he understood that this tall character was dominant over the dwarf factor, and that is why he called this tall factor as dominant factor, and the dwarf factor he called as recessive factor. This is what is stated in second law, that is law of dominance and recessiveness. The third law is law of purity of gametes, or it is also called as law of segregation of gametes. During gametogenesis, a factor enters the gamete singly without any mix, that is purely it enters one character. For example, if a gamete gets a tall factor, then only tall factor enters. It doesn't mix with any dwarf, or it comes in as pure one single character. There is no half tall and half dwarf. So it might seem very obvious to you, but then in those days these kind of principles were not known. So without any mix, when the factors enter into a gamete, it is called as that is what he stated, and called it the law of purity of gametes. By dihybrid and trihybrid studies, after doing these were the monohybrid studies. So when they ask question on first law of inheritance, then you should write about the monohybrid studies. When they ask question on second law of Mendel, then you should write about the dihybrid and trihybrid studies. So by dihybrid and trihybrid studies, he gave law of independent assortment. What is law of independent assortment? Each factor will enter the gamete independent of other factors. So one factor, if it enters gamete, it is not joined. So as I said, purely it enters in the third law. But in fourth law, that is law of independent assortment, only the character singly it enters. It is not bundled up with any other factor. For example, if tall enters, it is not bundled up with round or wrinkled seed coat. It can pair up with either round or wrinkled, but it is not packed together with a single uh, seed coat color, or for that matter even the flower color also. A tall plant can also have a violet flower. A tall plant can also have dwarf flower, a violet flower. So so that is what is law of independent assortment. Each factor enters into gamete independent of the other factors. So we'll come to critical analysis of the Mendelian factors. When you criticize, when you understand what is first law, then in that case we come to understand that Mendel knew only monogenic inheritance. He did not know polygenic inheritance that we know of now. Mendelian genetics talk about one character and one gene or factor that he called. One factor gives out one character. That is, a tall gene will only give rise to tall character, and a dwarf gene will only give rise to dwarf character. That is what he understood. But Nelson L. So while studying kernel coat color in maize, he said that more than one gene are required for expression of a character. So sometimes, so not all the times, that it is always one gene, one character, but most of the times, one gene is required for expression. More than one gene is required for expression of a character. That is, for example, when you take human beings, uh, if you think that gene A and gene B given by capital A and capital B gives melanin pigmentation, then presence of dominant AB on a person makes him very dark. On the other hand, if he has capital AB and small ab, small ab codes for white color, so he will not be so dark. He will be a light colored individual. And if he has AB and AB, that is small ab and small ab, then he will be purely white. So this I could have shown you in the blackboard, but okay. Uh, so when you try to understand, so capital AB gives rise to a very dark individual, and small ab gives rise to a white individual, and capital AB and small ab gives rise to a intermediate color, that is a fair skinned person, not so dark and not so light. So this kind of polygenic, because there are two genes giving rise to this melanin pigmentation, so was not understood by Mendel. So based on these experiments, if you do the studies, then you get a different ratio, and this different ratios are known as modified Mendelian ratios. So they are similar to dihybrid studies of Mendel, where he studied two characters, like for example tall and round. dwarf and wrinkled. But here we are studying only one character, but yet we are getting a similar dihybrid studies, but not same in fact, because in dihybrid studies Mendel got 9 is to 3 is to 3 is to 1 ratio. For example, if we take tall and wrinkled or round seed coat, then tall and round seed coats are dominant, and tall and round were found in 9 out of 16 F2 generation. F2 is the second filial generation, and 3 were found to have tall with wrinkled seed coats, and out of 16, and 3 out of 16 had dwarf with round seed coats, and only one among the 16 was uh, both were recessive factors, that is dwarf and wrinkled. But when you do in polygenic, that is when more than two genes are affecting one character, even if you study one character also, you will get a ratio like 1 is to 4 is to 6 is to 4 is to 1. So here also you get F2 generation 16 individuals. The first one will be pure dark, that is he will have A B capital on both sides, from mother side and the father side. But on the other hand, 4 individuals will have capital AB from father's side and small ab on mother's side. So these are not very dark, they are just dark. The first one individual is very dark, the next 4 individuals are dark, and the 6 individuals are called as mulattoes. They contain 50% dark from father and 50% dark from mother side. So they are mulattoes, where they have capital AB and small ab, capital AB and small ab. In case of first one, they had capital AB, the second individual will have capital AB, small a and capital B, and the third, that is 6 individuals that are mulattoes, will have equal half from father and equal half white from mother, that is capital AB and small ab. And when you go for next 4 individuals, that is 6 is to 4, you get one single from father, that is capital A, whereas all the other B and AB will be small. So single dominant factor giving rise to melanin pigmentation. That is, again, they are not very dark or not very light also. They are fair skinned individuals. And the last one, the single individual, that is 1 out of 16, will be small a and small b and small b, that is very fair individual. So hope I made myself clear, because lack of blackboard actually is killing me here. So 1 is to 4 is to 4, 1 is to 4 is to 6 is to 4 is to 1, that is the ratio. So the first individual in that ratio, 1 is to 4 is to 6 is to 4 is to 1, the first individual has all 4 dominant genes. So all the 4 will be producing a lot of melanin, and hence the individual will be very dark. The next, that is 4, these 4 individuals will have 3 genes that are producing melanin and 1 gene which is not producing any melanin, and that is why he is not so dark as the previous person. Then the next 6 people have equal black characters from father's or mother's side and equal white characters from father's or mother's side. And since they have 50%, that is 2 genes producing melanin pigmentation and 2 not producing, they are even more fairer, or even half black and half white, and these people are called as mulattoes. Then the next subsequent 4 will have only 1 gene that is producing black, and 3 genes are not producing any other black, and hence they are even fairer than the mulattoes. And when you come to last individual, he has no gene that is producing, that is all the 4 genes are non-melanin producing, and that person is a fair individual. So by this you understand that 1 is to 4 is to 6 is to 4 is to 1 is very different from 9 is to 3 is to 3 is to 1, that dihybrid studies. So this is what the Mendel missed out. He never understood that one character can be governed by many genes. Further, the second thing that Mendel failed to understand was the role of environment. A person's character, for example, stature, depends on nutrition also. So nutrition is what is outside environment. It is not inside genetic makeup. So a person can be dwarf due to less nutrition. So this factors Mendel did not give. He understood that characters are purely genetical, but rather characters are not purely genetical, but phenotype expression. Phenotype is how they look, the looking of the character outside. The phenotypic expression depends on genotype. It also depends on environment, and plus it depends on interaction of genotype with environment. So hence PE equal to GE, that is genotype expression, plus environment, plus interactions between genotype expression and environment. So that is actually the expression of phenotype. Mendel talked about only few characters, and those characters are called as Mendelian characters. These Mendelian characters are not affected by uh, the, the environment, and that is what he was very lucky that he had those characters that were not affected by environment, and hence he was very lucky to get Poisson sativum, and that is why maybe the Poisson sativum is also known as Cinderella of genetics. Now coming to this third one. The first one was polygenic inheritance. The second one is role of environment. The third one is what he did not sufficiently explain was multiple allelism. Now, according to Mendel, when he talked about the stature, for example, the character stature, he said there are only two factors governing it. One is tall and one is dwarf. But that is not always the case. Sometimes one character will have more than two, or even four, three, four, five different types of expressions. So that is called as multiple allelism. For example, if you take blood groups, human blood groups, as you all know, are three types. One is A, or they have three characters: A, B, and O. A, B, and O. A antibody, B antibody, and no antibody is zero antibody, that is O. Now there are three different characters, and based on the combination you can have any genotype. For example, it can be A0, it can be B0, or AB, or OO. So that is homozygous and heterozygous. But how many characters are forming here? How many factors are there? That is, alleles are there. There are three: A, B, and O. So Mendel only recognizes only two, where here we have three. There are many such characters which have many different alleles, and that is called as multiple allelism. For example, the coat color of a cat has 4 alleles. The coat color of a dog has 4 alleles. So you get the different colors of the dogs based on different combinations of these alleles, and this he did not explain. So Mendel consciously chose only those genetic traits which are represented by 2 alleles for his inheritance experiments. The fourth one, the first one was polygenic inheritance, the second one was role of environment, the third one was multiple allelism, and the fourth one is pleiotropic interaction. Pleiotropic interaction is just opposite to polygenic. In polygenic we see that many genes give rise to one character. Here it is the other way around. One gene gives rise to many characters. This is antithesis of Mendel. This is antithesis to Mendel. Mendel said one gene, one character. But here, one gene, many characters. For example, phenylketonuria. Now you take a disease called phenylketonuria, which happens due to one single genetic chromosomal defect. But this single chromosomal defect not only causes accumulation of phenylalanine in the blood of the person affected by phenylketonuria, but it also causes mental retardation. In addition to mental retardation, it also causes high arch palate. It also causes widely spaced incisors, uh, and it also causes excessive sweating. Now, one gene should give rise to only one trouble. If there is wrong with one gene, it should only give rise to accumulation of phenylalanine in the alanine, that is amino acid, in the blood of the individual. But you are seeing widespread effects such as mental retardation. Then you are seeing widely spaced incisors. Then you are seeing excessive sweating. So all these things show that one gene has many effects. One gene has many effects, and this is called pleiotropy. And pleiotropy was not understood or not given by Mendel. So, coming to the criticism of the second law. So, second law states that law of dominance or recessive. We have already understood what is dominance and what is recessive. If you, whatever the character you get in first generation, that is first filial generation, is dominant character. The second filial generation will have the character which is missed, which is not seen in the first generation of true breeds, pure breeds, is called recessive character. So Mendel said about dominance of single character. He said one single character is dominant, but but it is not always the case. For example, you see incomplete dominance. What is incomplete dominance? Incomplete dominance is, for example, if you see the flower of snapdragon. The flower of snapdragon is red and white. It is homozygous for red, it is homozygous for white. But if you find a heterozygous, for example, 2 red will give very red color, 2 white will give you very white color. But if you have a red gene from red flower and white gene from one flower, that is red and white, this will give rise to pink flower. It will not be either red nor white. So neither red is dominant nor white is dominant, but both are dominant. So you get half of its expression from red color, half of its expression from white color. Mind you, it is very similar to polygenic inheritance. Polygenic also we have seen. For example, one character, now different genes are giving rise to color there. So like in polygenic inheritance, A gene is giving equal amounts of melanin as B gene is giving. But here it is only one gene and it is having a expression. It is dominant. It is also giving rise to color. The other recessive gene also is giving rise to color. Hence we cannot say this is dominant or recessive. Hence we call it as incomplete dominance, or it is also called as equal dominance. So equal dominance, incomplete dominance. Now this is the criticism of second law of that is law of dominance, because in nature they are not very simple. It is not only dominant or recessive, but rather you see incomplete dominance also. Similar to that is another concept, that is codominance. Codominance. For example, if you see our ABO blood group, coming back to ABO blood group again, ABO blood group show codominance. A person in ABO blood group, both A and B, both are dominant. If a person has A0, he is A blood group. A person having B0 is B blood group, and a person having A and B will be AB blood group. So now you understand that both characters are coming and sharing dominance. Both of them are dominant. In the earlier part we saw both of them collectively become dominant, but here both of them are fully dominant. There it is only half the expression of dominance, that was shared dominance, but here you have co-dominance, that is both of them are dominant. And on top of this also was not understood by Mendel, and he did not give. Later scientists, they understood and they gave these terminologies. And the other one is epistasis. What is epistasis? Epistasis, or it is also called as inhibition. For example, some genes, though dominant, for example, if you talk about one gene gives rise to one character, and if that gene is dominant gene, it gives rise to the dominant character. But that is not always enough for a character to be dominant. There has to be another recessive gene somewhere which has to provide expression to this dominant gene. So on other location, a recessive gene on other location will in fact give rise to dominance to this gene, in and help it to produce a dominant character. If that recessive gene is not there anywhere elsewhere, this gene cannot be produce the dominant character. That is what is epistasis. So these are the basic criticisms of this should be sufficient for your exam. Usually Mendel's laws of inheritance will come for 20 marks questions or 15 marks question. uh, if they ask you first law, then this part is sufficient. What we have talked, the first 1 2 3 laws and the critic till now, so the first law they will ask you like this. The second law, if they ask you, then you have to tell them about dihybrid studies, trihybrid studies, law of independent assortment, and then critic of third and fourth law. That there are two critics here, third and fourth law. The first one is genetic linkage, the second is intranuclear inheritance. So in fourth law, in third law, he says, for example, law of segregation of gametes or purity of gametes. This is the only law which holds good in all conditions, in all, whether the character is dominant or not dominant, dominant, co-dominant, or it could be whatever the character. But then there is purity of gametes. One character will just enter one gamete. So this holds good in all cases, and hence there is no criticism for third law. But the fourth law, when you come, fourth law is law of independent assortment. Law of independent assortment was given after dihybrid and trihybrid studies. So law of independent assortment, it states that each factor will enter the gamete independent of the other factors. For example, if a tall factor is entering, so only tall factor will enter. It will not come up bundled with a round factor or a rot or a violet color flower factor or a green pod color factor. So this person is independent of himself. This character is independent of himself. This allele is independent of himself. It just enters as a whole, and it doesn't come with another character bundled. So that is what is law of independent assortment. But it is not always the case. For example, you see genetic linkage, something known as genetic linkage. When you see HLA, human leukocyte antigen, so you have a lot of genes contributing to HLA antigen system. So they all enter in toto, they all enter as in package. There is no crossing over during that phase. I will talk about crossing over in subsequent classes, what the crossing over means when we talk about the meiosis and all those things. But during the crossing over, you find the entire piece and parcel of the genes come as bundled. So it is against the Mendel's laws of inheritance, which states that the characters cannot be bundled. But here you find they are bundled, and this bundling of characters is known as genetic linkage. The second part is extranuclear inheritance. When he said only one character comes in, but and it is always later it was understood that this comes from nuclear. The nucleus contains the DNA, and from there it comes, the characters are inherited. But then DNA is also present in mitochondria of the cell. This is called as cytoplasmic inheritance. Now if you understand that ovum of the mother contains all the cell organelles. The sperm of the father contains only the nucleus. It doesn't contain any cell organelles. And when they fuse, so the only the half of the nucleus of the zygote is given by the father, whereby all the rest of the cell organelles that are present in the zygote come from the mother. And so it is the same for even mitochondria also. Mitochondria also comes from mother's side. And this mitochondria contains certain a small bit of genome, and that genome is also inherited, and and a son almost gets all his genes, or a daughter gets all his mitochondrial genes from only mother's side and not from the father's side. Only maternal inheritance is seen in extra nuclear inheritance. So there are a set of, in humans you have many mitochondrial diseases. They are a class of diseases, many mitochondrial diseases that affect the muscles and eye. So they come from the mother's side. They were first discovered by Carl Correns in 1808, and who discovered, and later it was discovered that the genetic material is found in mitochondria and not in the cytoplasm. But it is always called cytoplasmic inheritance. So I'll stop this right now, and hopefully we can have some other class sometime.